paper at Hanel was testing the efficacy of green Rooibos extract to counteract the effects of obesogenic feeding on adiposity and rat adipose stem cells.
I'm a stem cell biologist with a particular focus on using the stem cells within adipose tissue, fat tissue, as a model to study the development of fat cells and then also obesity along with that, and I will show you a little bit later on how that ties in with Rooibos. So what I'm going to do today is actually explain to you the model that we'll be using to test Rooibos in the context of obesity. Okay, so just to give you some background, South Africa is an obese nation. When we look at overweight, that's a body mass index of greater than 25 kilograms per meter squared. The data, recent data shows that 40% of South African men, a quarter of our children, and a whopping 70% of our women fall into the overweight category, and 40% of our women fall into the obese category. That's a body mass index of greater than 30 kilograms per meter squared. The adverse health consequences and comorbidities associated with obesity are numerous, and given the number of overweight and obese people in our country, these comorbidities are set to engulf our healthcare systems. The Global Burden of Disease study that was conducted by the Harvard School of Public Health, looking at data from 1990 to 2015, indicated that the disability-adjusted life years, which includes then also the number of deaths associated with obesity, has been rising quite steeply in recent years. And this is because obesity is associated with these comorbidities, diabetes, stroke, hypertension, cardiovascular disease, and even certain types of cancer. And the increase in disability-adjusted life years are then also telling us that more and more people are suffering because of their obesity, even if they don't die from it. But we know all this. So if we know all this, why do we still have this frightening prevalence of obesity in our country? Why can't we just all be lean, or get lean, or stay lean? Sure, there are individuals who have had spectacular results with particular diets, and we read about them in the magazines, but I think it's clear on a population level that managing body weight through diet and lifestyle changes is just not as simple as it sounds. Metabolic surgery is available for morbidly obese people, but we simply cannot operate on every obese person in this country. We've also been largely unable up to now to formulate weight loss drugs that A, actually work, and B, don't have horrible side effects. So we don't have an easy answer that will work for everyone. And after working in this field for a number of years, I believe that this is because we don't really understand how fat tissue works. We know on a very basic level that our fat cells, adipocytes, store excess calories in the form of fats and sugars until such time the body needs it, but we have a really limited understanding of the factors governing the storing and the burning of fat. And if you can't really understand the problem, then we have little hope of solving the problem. So there are, in my opinion, two reasons why we don't have a proper understanding of how fat works. One of the reasons is that fat is seen as the enemy, something that has to be eradicated, right? But that slants the way that we actually look at fat. And we only study things that we hope and think may reduce fat or fat cell formation. But the truth is that fat is an essential organ in our bodies, and it performs functions that we cannot do without. And it is in our best interest to understand how healthy fat works so that we can understand what happens when it goes wrong. So, what does fat do that is so fantastic? So it protects us from the outside, it also protects our internal organs, and it keeps us warm. It's a crucial store of excess calories, especially in the form of fats and sugars. If those excess fats and sugars have nowhere to be stored, they remain in the bloodstream. And this results in elevated blood glucose, elevated blood lipid levels, which leads to diabetes, high cholesterol, atherosclerosis. And we know this because people with lipodystrophy, people with an absence of fat, which may sound fantastic, they have the same metabolic abnormalities as people with too much fat. So we need fat. And then lastly, fat tissue is also an endocrine organ that produces circulating factors into the bloodstream, similar to hormones. And these factors are absolutely vital to maintain a normal metabolic status. And when we look a little bit closer, we see that not all areas of fat behave the same. Subcutaneous fat, which is the layer of fat directly under the skin all over our bodies, that is metabolically stable and it serves as the first safe storage space of excess calories. But once the subcutaneous fat is filled up, the overflow ends up in the visceral abdominal fat, in the boob, as we call it. But this is problematic because visceral fat produces factors that actually promote diabetes, insulin resistance, systemic inflammation, atherosclerosis. So one of the hot topics in the field at the moment of obesity research right now is actually the expandability of subcutaneous fat. How do we actually program our bodies to store fat only in the subcutaneous fat and prevent it from going into the visceral fat? We don't have the answer yet, but it is a very hotly debated topic. So for this reason, it might then actually not be actually a bad idea to suppress the formation of new fat cells, especially in the subcutaneous fat. I mean, it sounds like such a simple solution. Just find a way of blocking the formation of new fat cells and we'll solve obesity. There, done, bye. But it doesn't actually work that way. Fat tissue needs to perform its storage function. And when no new fat cells are being formed, then the old existing fat cells just have to get bigger and bigger, and that causes adipose tissue dysfunction, and that just takes you right back to diabetes and blocked arteries. The other problem is that fat tissue that used to be obese and it is now lean is different from fat tissue that's never been obese. Obesity leaves behind an obesogenic memory inside the fat tissue that causes adverse long-term metabolic consequences and can actually be just as detrimental to your health. And it can also relapse into obesity quite easily. So this is all a lot to take in. But why am I telling you this? It's so that you can understand that we all need a healthy amount of healthy adipose tissue to stay healthy. And therefore, the fight against obesity cannot be reduced to a simplistic aim of reducing body weight and suppressing fat cell formation and fat content. And any diets and purported weight loss products need to be evaluated for their effect on fat tissue and their effect on fat cell development and function before we can decide whether it's a good idea to use it or not. Now, the other reason why we have such a poor understanding of adipocyte biology and function is because the models that we choose to study adipocyte biology with are really inadequate. We don't currently have the tools to look inside the fat tissue of a living person. Maybe one day we will. That would be nice. We can take a small biopsy of fat tissue from people who are willing to hold still for long enough, but we can only reach the superficial fat that way when we really need to be studying the visceral fat, the abdominal fat, which is the more pathological one, or at least be comparing the subcutaneous and the visceral fat. So that's not really the way to go. So we have to make do with other models, other representations of adipocytes that we grow in cell culture. But the most popular adipocyte cell line in the world, even to this day, was generated from one mouse in 1974 and has been growing and been passed around the world ever since. Another cell line that we also use, SGBS cells, was generated from humans with a genetic adipocyte overgrowth disorder. And although these cells are very convenient to use, and although they give us a marvelous representation of what happens when a non-fat cell turns into a fat cell, or the cellular events associated with that, we cannot mimic any of the systemic whole body effects on fat tissue this way, and we cannot track what happens when an otherwise healthy person becomes obese. We can directly apply compounds such as weight loss drugs into cell culture, but when we take medication, it's metabolized through the digestive system and the liver, so what ends up in the tissue is not necessarily what you swallowed originally. So direct effects that are observed in cell culture might not be representative of what happens on a systemic level in the body. We also cannot mimic any of the physiological factors that we know affect our adipose tissue, such as age, gender, pregnancy, diet, exercise. We cannot mimic any of that in cell culture. So we have to find another way of studying the role of these factors in the development of fat and then in the progression of obesity. So, since the early 2000s, some researchers around the world have adopted a different strategy, a different model for studying adipocyte biology, and these are called adipose stem cells. As the name indicates, they are the stem cells that reside within fat tissue and they develop into new adipocytes. They also contribute to the metabolism of the fat tissue and also the systemic metabolism by secreting circulating factors into the bloodstream, and they also become dysfunctional during obesity. So they can therefore give us a much clearer picture of what happens within fat tissue as obesity develops and progresses. But the true power of these cells is that we can use them in an in vivo, ex vivo study design. So that means instead of trying to mimic whole body situations in a cell culture dish, which we really cannot do, instead of treating the cells, we treat the whole organism, living organism, in vivo. We remove the stem cells afterwards. We study them outside of the body, ex vivo, and see how what we did in vivo changed the biology of the cells. Okay, so last year I published this review called Adipocyte Biology. It's time to upgrade to a new model. And this review was part opinion statement and part survey of the field as it stood at the time. So the opinion statement part basically argued that we should all move away from using these outdated cell lines and all start using adipose stem cells as a adipocyte model. The survey of the field was basically to determine who has studied and who has published what in these cells. So I looked at all the studies published around the world using adipose stem cells as a model for adipocyte development. And to my utter surprise, I found that while many studies compared adipocytes from lean and obese individuals, people and animals, very few studies prospectively looked at the changes in adipose stem cell function while the subjects were on an obesogenic diet. To be exact, only 7 studies from around the world have ever looked at the changes brought about in the adipose stem cells when exposed to a obesity-inducing diet. And up until the middle of last year, when this review was published, exactly 0 studies have ever examined the effect of any botanical products or any weight loss drugs on adipose stem cell function. No one has looked. So I would like to change that. So this is my study design. It's already underway. So we're doing this in male rats. We have 3 groups of rats: the lean control diet, where they just eat their little pellets, a high sugar diet, and a high fat diet. We then feed these animals for 10 weeks, and then after 10 weeks, the diet groups are divided in half. Half of the animals start getting a green Rooibos extract daily for 7 weeks. While the other half don't, and then at the end of the feeding period we sacrifice the animals and then we harvest everything: blood, heart, liver, pancreas, kidneys, guts, testes, muscle, bone, everything. There's an empty husk left when we're done. I harvest the fat from the subcutaneous and the visceral sites, and I isolate the adipose stem cells, and then I grow them in culture, and then we stimulate them to turn into mature fat cells. And then from these cells, we will then analyze their gene expression, their protein expression, the factors that they produce, which would then end up in the circulation, the types of fat that they accumulate intracellularly, and then we correlate that with what's happening in the serum from the same animal to see whether there is correlation between what happens in the adipose stem cells and what happens in the whole body. And by measuring all these things, we can then determine A, what goes wrong during diet-induced obesity, what is the nature of the dysfunction that is induced, and B, whether Rooibos can be effective at reversing these dysfunctions. So the advantages of my work will be twofold. First of all, the study will generate hard scientific data on the possible effects of Rooibos on the development of adipocytes and on the function of adipocytes in fat tissue. So here we really have to commend the Rooibos Council for supporting research of this kind. But as an added bonus, in the process we will also learn a lot about the effects of high sugar and high fat diets on adipose stem cells. And we will become a member of the very small group of scientists worldwide who are studying the effects of diet on adipose stem cells. But it's also important to keep in mind that although this is a project on Rooibos and it's funded by the Rooibos Council, it's important to think bigger and look wider. This type of study design can be applied to any diet or product or drug that we think may affect fat tissue or may be helpful in treating obesity. And it can also be used to study any physiological conditions such as diet and age and pregnancy on adipose tissue. So I look forward to maybe one day be able to study all of these things. I would just really like to acknowledge the following people, all my collaborators. We're quite a lot of people working on this at the moment. The Rooibos Council, which is the main funder of my study. The National Research Foundation, and also the South African Sugar Association for co-funding. Yes, that's ironic. And then a special mention must go to the Stellenbosch University Faculty of Medicine and Health Sciences, where I'm based. We started feeding the animals in October, and we isolated the first stem cells in February. But the equipment that we need to keep stem cells alive and growing don't work during load shedding. And our faculty management was really at the foresight a few years ago to install generators that can actually power the entire campus during outages. And without that, this study would have absolutely irretrievably collapsed a few weeks ago. So they really deserve a special mention. So thank you for listening.
Are we working off a theory at the moment regarding what effect Rooibos is going to have on adipose stem cells?
In the old days, the technology was such we could only measure one thing at a time. So you had to sort of go in with a particular, very well-defined hypothesis. And as things progressed over the past 2 decades and so, we now have the tools to actually measure hundreds or sometimes thousands of things at the same time.
Yeah.
Without a clear hypothesis going in. And in the past, it was really frowned upon. It was called a fishing expedition. But our attitude towards that has really changed, and now it's called unbiased research. Which is really great, because yes, you can't just sort of blunder around, I agree. But sometimes it helps to just look at a whole lot of different things, see what pops out, and then from that, develop a more targeted hypothesis. We don't know, because like I said, no botanical product has at all been tested in the world ever. But given that there's a lot of information out there about the other metabolic benefits of Rooibos, it would be difficult to imagine a situation where it wouldn't have an effect also on the metabolism of fat tissue.